Uh, all right. Good morning, everyone, and welcome to the Midwest Bioinformatics Showcase. Thank you for joining us today. My name is Arun Sitaram, lead bioinformatics scientist at Rosen Center for Advanced Computing, Purdue University, and along with me, Haley Carter, Northwestern Research Computing Data Services, Northwestern University's Research Computing and Data Services. Together, we organize this series. Today is actually our final seminar of the spring 2026 semester season. We have had 8 fantastic talks this spring covering everything from digital pathology and foundation models to pangenomics, agent-based modeling, methylation sequencing, and multiomics integration in cancer. It's been a great showcase of the computational work happening across both universities, and I want to thank everyone who has attended, asked questions, and helped build this community over the past 2 months. As always, today's session is being recorded and will be posted on our YouTube channel and on our website, midwestbioinformaticsshowcase.com. A few quick housekeeping notes. Please keep yourself muted during the talk. We'll take questions at the end. You can either raise your hand during the Zoom uh, meeting or drop a question in the chat and I'll read them out. The talk will be about 35 to 45 minutes, and at the end, we'll have Q&A. With that, let me introduce today's speaker. Our speaker today is Yueming Liu, a PhD candidate in the biostatistics track at Northwestern University, where she's advised by Professor Denise Coltons. Yueming's methodological research focuses on probabilistic graphical models and Bayesian approaches for high-dimensional biomedical data, with particular emphasis on capturing dependency structures and subject-specific network variation. Her work bridges the statistical methodology and applied biomedical research. She develops methods that integrate covariates, molecular features, and clinical outcomes to improve interpretability in complex biological systems. On the applied side, she collaborates on studies using epigenetic and proteomic data to investigate mechanisms underlying metabolic and cardiopulmonary outcomes. Today, will present our work on evaluating ancestry adjustment methods in multi-ancestry epigenome-wide association studies. Yueming, the floor is yours.
Hi, I'm gonna share my screen. Everything looks good? Okay, that's great.
Looks good. Thank you.
Good morning, everyone, and thank you for the opportunity to present my work today here. My project focuses on evaluating ancestry adjustments in multi-ancestry epigenome-wide analysis, or EVAS. In EVAS, we test associations between DNA methylation levels and phenotypes of interest. It has become a powerful tool for us to investigate molecular signatures that is related to complex trees and phenotypes. So here is the figure demonstrates the DNA methylation. So DNA methylation at CpG sites may alter gene expression, thereby impacting biological pathways and contributing to phenotypic variation. One main challenge in EVAS is that DNA methylation levels can vary systematically across different ancestry groups. So if we did not adjust it for this factor properly, it can lead to inflated genetic factor and spurious associations. So it is there is a similar concept in GWAS. In GWAS ancestry correction, ancestry is commonly adjusted using genetic principal components. Well, but in practice, when so most cases, in many cases, we don't have access to genetic data. So there is a need for us to find out a way to do ancestry adjustments when we only have methylation data. So the commonly used method is epistructure. It is a reference-based method for ancestry adjustments in EVAs, which is developed by Rimani's group. So here the key concept is that the CpG sites with strong correlations with local genotypes, cis-SNPs. These are selected as reference. So based on the reference set of CpGs, PCs are extracted to account for ancestry structure. So the one limitation here is that the canonical epistructure reference sets were derived from the cooperative health research in the region of Augsburg, the CORA study, which only consists of individuals of European ancestry. So here we wanna derive new reference set that is based on a more diverse cohort, the Hubble cohort here. The hypoglycemia and adverse pregnancy outcome Hubble study. It is a sample size about 3,000 individuals. The methylation platform is using Illumina 850K array, which is capture broader CpG coverage. The ancestry composition, the Cora is primarily European ancestry. The Hubble is a multi-ancestry one, and it is more diverse. The implication for EVAs is that we think that the epistructure PCs based on the Hubble reference set may better capture underlying ancestry structure in diverse cohorts, and therefore broader CpG coverage enhance ancestry resolution. Here is some idea about how do we construct those reference sets. We use paired genotype data to identify ancestry informative CpGs based on CpG cis-SNPs R-square, constructed reference set based varying thresholds, which means that in simple word is that we are trying to find out a couple set of CpGs that have higher correlation, a stronger association with the genetic cis-SNPs, because a previous study shows that those CpGs are capture more ancestry information. The target phenotypes used in the EVAs are reveal studies newborn and positive cheese, birth weight logs called C-peptide, and some skin folds. We evaluate our EVA's performance by a couple metrics. The first one is ancestry resolution, and genomic inflation in EVAs and phenotype-associated CpG discovery. That is more focused on the biological interpretability, that the goal enrichment and treat enrichment. So here is the construction procedure. So ancestry, we talked about it earlier. So ancestry-related signals in DNA methylation are primarily driven by widespread correlation between methylation levels and underlying genetic variation. Since the majority of the reported CpG SNP associations involve cis-located SNPs, we focus exclusively on cis-SNPs to capture the genetic components of each CpG. And then we fit a regularized linear regression model, LASSO, for each CpG using cis-SNPs as a predictor and evaluate the performance using cross-validated linear correlation, R-square. So based on the R-square, we have a couple thresholds. We are picking 3 thresholds here, the 0.3, 0.5, 0.7. They are denoted as HAPO 30, HAPO 50, and HAPO 70 in the following analysis. In the CORA study, their original study using a cutoff of 0.5. So here's the overlap of CpG sets across reference set for epistructure. Before we do the EVAs to test the adjustment performance, we also need to adjust for cell type. So cell type composition is another key confounder in EVAs. DNA methylation is highly cell type specific, and copula contains a mixture of immune and progenitor cells. Variability in cell proportions also can lead to spurious associations like ancestry confounding information. So we are using refactor. It's a reference-free method that uses PCs derived from sparse PCA to adjust for hidden cell type structure without requiring known cell type profiles. So here, based in addition to epi-structure PCs, we are having couple another refactor PCs that capture the cell type information. So here is the model for the EVAS modeling. For each phenotype, we fit separate linear regression models at each CpG set. We adjusted for child sex, 7 cell type principal components, which stand for 7 major types of cell types in practice, and also maternal covariates and ancestry PCs. So here we, in the analysis, we systematically iterate the model with the number of PCs, ancestry PCs from 1 to 10, because we want to investigate how does the adjustment performance changed by the number of PCs included in the model. So here is the 3D visualization of epi-structure PC scores. Here is the more validation purpose. On the z-axis is the PC score for the top 1 genetic PCs, and on the x and y axis are the top 2 epi-structure PCs. So we can see that there show some pattern here. We also color the points by maternal self-reported race ethnicity here. So we point that even though this may not fully capture newborn genetic ancestry, but it remain sort of informative, and we can treat it as a reference. Here we can see there is a strong alignment with the top 1 PC score generated from the genetic data and top 2 PC generated from epi-structure PCs. The left-hand side is based on the core reference set. The right-hand side is HAPO-IS30. Here is HAPO-IS50, RS70, so they are pretty similar. So the reason we present the figures here is that it means that those epi-structure-based procedure for ancestry adjustment is pretty robust. And here is a heat map for the epi-structure PC loadings. On the rows, there are different CpGs, and on the columns, each column represents a PC here. Different color means different reference sets. We can see the right ones are PCs comes from the Cora reference sets. Here, those blue ones are all for the HAPO ones. The white blank here means that the CpG is not present in the corresponding reference set. We can see that there are some darker color shown here, which means that if there is a darker color, it means that those CpG are more, we can say that it is more informative for the ancestry information. But we can see that here for the HAPO ones, there are also some pattern here, which means that which may suggest that the HAPO reference set may capture some ancestry information, while the Cora cannot. We evaluated the EVA's performance using genomic inflation factor. The lambda measures how much the observed test statistic deviates from their expected values under the null. For each CpG set, the null means that the methylation at this CpG is not associated with the phenotype. So since the vast majority of the CpGs are expected to have no true effect, so this now provides a good approximation for the bulk of the genomic inflation. So the interpretation is that lambda greater than 1, the generalizes that there is an inflated lambda and there are some residual confoundings. If lambda is smaller than 1, it might be too conservative. There is some overfitting or over adjustment. Here are the lambda values line plots for 3 phenotypes. We can see that for all the phenotypes, increasing the number of ancestry pieces generally reduce the genomic inflation across all phenotypes. So in the downstream analysis, for each reference set, we only keep the model that's including top 10 ancestry pieces. Then another point is that we can see sometimes, especially for called log C peptide, the haplo-IS30 sometimes even achieve a lower lambda than the genetic PC. So which indicates that the methylation-based PCs generated from haploreference set may capture some ancestry information on the methylation level, while genetic PCs not able to. Here is also some QQ plots of EVA's results. And here I have put the lambda values in the figures also. So in general, the blue ones are haplo-IS30. IS30 is the best performance one. So in the downstream, we will focus comparison between Cora and haplo-IS30. You can see for birth weight, the haplo one have the smallest lambda value. And if we take a look at if we zoom in the Q plots in the lower range of the distribution, if we take a close look at the blue and gray lines, we can see that the haplo is able to better control the inflation in the bulk of the distribution. And also we can see that in the tail of the distribution, there are some more significant points here. So it means like it provides improves overall calibration of EVA's test statistics while preserving trait association signals. So the calibration is reflected in this region, and the trait associated signals is reflected in the tail behavior. And of course, across all trees, the observed test statistics shows moderate deviation from the null expectation, which is consistent with the presence of both true associations and residual confounding typical for large-scale EVAs. For the overlap of significant CpGs identified by Hubble as at Cora, here we're also adding genetic PCs, significant CpGs here. We can see that across all 3 phenotypes, there is a substantial overlap across all those adjustment strategies, which indicating a common set of robust associations. For the sum scaffolds, there is remarkably smaller set of significant CpGs identified, which is as expected because sum scaffold is more tissue specific. Some of the biological information may not be captured very well on methylation level. To better evaluate our ancestry adjustment strategy, we perform functional enrichment, genealogical enrichment, identify biological pathways that are overrepresented among phenotype-associated CPGs. In those kind of enrichments, each CPG is mapped to a gene based on its genomic location. The selected CPGs are converted into a gene list and tested for enrichment for goal terms, revealing overrepresented biological functions. Here the input selection is critical. Those kind of enrichment is highly sensitive to the size of the input CPGs. Therefore we selected the top 1,000 CPGs with buffer-only corrected p-value smaller than 0.05. I would like to point out that in this Venn diagram, the significant CPGs is defined as p-value smaller than 0.05 after buffer-only correction. So like the genetic PCs and HAPO identify more than 1,000 for birth weights. Well, for CORA1, for some phenotypes, for a couple of phenotype, it is smaller than 1,000. So I would like to explain why we are picking top 1,000 CPGs. If for each reference set we use a threshold that's it is significant, which means that for each reference set the number of the input set is different. But those kind of enrichment is very sensitive to the input size, so we want to make the number of CPGs fixed across reference sets. And if we are putting only 500,000, the number is too small to identify any informative pathway. So therefore, we selected the top 1,000 CPGs as the input for goal enrichments for all the reference sets. Here is a scatter plot for the top enriched goal pathways. The left-hand side is for HAPO R30, the right-hand side is for CORA. The points that are across this dashed line means it is significant at a threshold of FDR smaller than 0.1. The CORA does not able to identify any significant pathways here. The HAPO captures something here. So these terms show stronger statistical significance under the HAPO R30 reference. HAPO R30 uniquely identify top-ranked immune regulation pathways, like the lymphocyte-mediated immunity, regulation of lymphocyte-mediated immunity. These pathways have been linked to gestational immune regulation, fetal adipose tissue development, and metabolism. Here's are some readout for the top enriched goal pathways called C-peptide. So for this one, it generally is weaker than the phenotype birth weight. But overall, we can say that the HAPO R30 tends to show a stronger ability to capture biological relevance pathways here. So here I'm listing some key immune-related pathways. So leukocyte-mediated immunity. So these are all important pathways for neurital adiposity related phenotypes. The EVAS catalog, so we also do some traits enrichment via EVAS catalog. So it is a database of CPG trees associations from published EVAS research, which is enable evaluation of the biological relevance of discovered CPG by checking overlap with known phenotype associations. We perform tree enrichment analysis using CPGs uniquely identified by HAPO and CORA reference sets. Trees enrichment results help categorize whether reference set specific CPGs are linked to trees relevant to fetal growth, metabolism, or immune regularization. So HAPO identified more trick-specific, while CORA capture more general in their biological relevance. So here is a bar plot here. The y-axis is trees, and here the number means that the number of studies we found in the EVAS catalog for those significant CPGs identified by those reference sites. So in general, in discussion, ancestry-informed reference set can influence the sensitivity and specificity of EVA's results. The HAPO one shows stronger enrichment of immune-related goal pathways relevant to fetal adiposity. It shows greater specificity in EVA's catalog trait associations, highlighting metabolic and inflammatory phenotypes. CORA identify more general or less biological relevant goal terms. So these findings suggest that reference set derived from ancestrally diverse cohort like HAPO may more effectively capture ancestry-associated methylation variations in diversity populations, enhancing both statistical power and biological interpretability. That's pretty much all my presentation. Thank you so much.
Thank you, Waiwei. That was an excellent presentation. We now have time for questions. So any of the audience who have questions, please raise your hand or just unmute yourself and speak up. Yeah, so please go ahead and ask your questions. If not, I'll go with my first question. So you selected the HAPO reference cohort. Was there like a specific? Why were you specifically? What drew specifically into this HAPO reference set? Were there other multi-ancestry methylation cohorts you considered, or this is the only one that's available?
Oh, that's the only one we have.
Oh, okay, that makes sense. So if someone is running a EBUS today, what would you point out? Point them to APO reference versus the original Cora-based epistructure. Is there a rule of thumb selecting one of them?
I think it depends. So if the cohort is also like the Cora is just a European population, so maybe I think the Cora may be more appropriate. If it is a more diverse cohort like HAPO, I would say HAPO might be able to adjust the ancestry information better because it's a reference CPG set based on a very diverse cohort. But because of the one limitation of our study is that we don't know how does the proportion of the populations can influence the reference set construction. So we would say that if they are able to, if they have access to genetic data also, and they also wanna adjust the ancestry information at the methylation level, I would suggest using a similar procedure as we done in Hubble here. If they don't have access to genetic data, I would say Hubble I-30 reference set may be a good alternative for the genetic PCs.
Okay, thank you. Looks like we have a question from audience. Jong Yoon asks, do you recommend using genetic PC instead of epigenetic PC to account for the population structure if the genetic data are available?
Actually, from our analysis, we can see here sometimes the epi PCs performs better than the genetic PCs. And I didn't put the figures for the goal enrichments and treaty enrichment for genetics data here, but we also have the similar analysis on the genetic PCs. The performance is pretty much like the Cora one. It is not be able to capture as much as about those strong biological relevance pathways here. So in terms of controlling the genomic inflation factor and also the biological interpretability, I would say in our study, the IP structure-based one is better than genetics one, but the relevant study about the genetic PCs and how does the DNA methylation capture the underlying ancestry information is remain to discover. So I would say maybe both works, but it depends on the situation. If we have both access to genetics, maybe we can try both and then do comparison.
Thank you. I'll go with one last question. Where do you see this going next? Like extending to other phenotypes or like cohorts or porting it to an epic array or something else?
So the reason we pick these three here, like first it's called C-peptide and some skin folds, because these three phenotypes are three well-studied ones in neonatal adiposity. So it is easier for us to validate our biological relevance here. So our analysis is based on the cord blood profiles. So I would say the study results may be limited by the cord blood profiling. Yeah.
Okay. Thank you.
All right.
I don't see any more questions, which means that that's all the questions we have today. Yueming, thank you very much for really compelling talk to close out our series. Before we wrap up the session, I would like to thank everyone um, um, who made this series possible. First, Haley Carter, who is our co-organizer from the Northwestern University, Nadia Landman and William Kath, both professors at Purdue and Northwest University. They were the reviewers of all the abstracts that we received, and they made the selection process, everything go smoothly. And then Katie Gunderson, who is our digital content specialist at Purdue University. Eric Adams, lead research operations administrator for education. And all our speakers who shared their work with the community. A special thanks to all the people who attended, whether it's one or all 8 sessions. Really, thank you everyone for attending the series. All the recordings are being posted to our YouTube channel and our website, midwestbioinformatics.org. We'll be sending out a short feedback survey in the coming days. Please keep an eye out of it, for it. Your responses will help us shape up the future of this series. So we plan to do this reoccurring every semester. So please give your feedback, and we'll make adjustments based on the your feedback. Um, thanks again, everyone. Have a great weekend, and thank you. I'll see you all in the next season of MBS.
Thank you.
Bye.